Good afternoon, everyone. I would like to thank the organizer for giving me the opportunity to present my data. My name is Massimiliano Bissa. I work at the Animal Model and Retroviral Vaccine section as a staff scientist. This lab is led by Dr. Vefa Francini, and we are located at the National Institute of Health in Bethesda in the United States. Let's start with some background. In 2022, we're estimating 1.3 million new HIV infections worldwide, and 80% of them occur via mucosal transmission. Unfortunately, antiretroviral therapy is still very expensive. Therefore, the development of an effective anti-HIV vaccine, even with a 50% efficacy, could meaningfully reduce the HIV transmission. The challenges to develop an HIV vaccine are many. HIV infect is genetically heterogeneous virus form, is often resistant to neutralization, and the fact that the vaccines induce antigen-specific responses via inflammation can be problematic because this could induce and recruit CD4 positive cells that express CCR5, and therefore they are target cells for the HIV infection. During the development of HIV vaccine, several trials in phase 2b and 3 were tested, in particular 7 of them, and the majority of them actually failed in proving any vaccine efficacy, except in the RV144. The RV144 proved a 31.2 vaccine efficacy, was tested in 60,000 volunteers, and was carried on in Thailand. The volunteers were either placebo or vaccinated with a prime boost protocol involving the viral vector ALFQA, which is an avipox virus expressing HIV genes, followed by a boost in which the ALFQA was administered in association with the GP120 protein formulated in alum. ALFQA is able to produce viral particles, and the clade used in this trial was clade B and AE. The studies identified that there are several immunocorrelates of protection for the RV144, in particular non-neutralizing anti-V1-V2 IgG correlated with the reduced risk. One of the hotspots for induction of antibody-dependent cellular cytotoxicity was the V2 region, the variable region 2 on GP120, and the ADCC also correlated with reduced risk. The polyfunctionality induction of these V2 specific T cells was finally correlated with reduced risk. So all these results point out of the importance of targeting the V2 and V1 region of GP120, which is actually on the apex of the trimer of the GP120 that is on the virion, and the V2 contains also the binding site for alpha-4 beta-7. We at the NIH, we developed a non-human primate model that is very useful and powerful to develop a new vaccine and to study the immunogenicity, and was developed by Dr. Francini at the NCI. This implied the using of a vaccinated macaque and controls following vaccination with a protocol that can last from 3 to 6 months. We analyzed the different immune responses, but we also exposed the animals to the SIV MAC251, which is the counterpart of the HIV, but for the monkeys, and we follow the infection, and we compare the infection on the control versus the one on the vaccinated, and as a readout of the vaccine efficacy we have the analysis of per exposure risk of viral acquisition. We also apply a system vaccinology to understand how the vaccination is inducing the immune responses with several type of technique. This model was actually very relevant to the humans. In fact, we recapitulated the results of RV144 and predicted the failures of the clinical trial HVTN702 and HVTN705, as well as confirmed the binding antibody as V2 correlated. And we published especially 2 papers in Nature Medicine 2016 and 2018 about this. We also use this model to compare priming, compare viral vector, compare adjuvants, and compare genetically modified GP120 to actually improve the vaccine efficacy and try to increase it. So we came at the end with a streamlined regimen with the use of DNA prime followed by 1-ALVAC and 1-ALVAC in association with the V1-deleted protein as a boost, as one of the best vaccine protocol. And both the DNA and the ALVAC are actually able to produce VLP, virus-like particles. We also use the system vaccinology to understand what are the immune responses that are actually correlating with increased risk of HIV acquisition. And what we identify in our study is that antibody responses against the V1 region was actually non-protected. And when we look at the structure of the GP120 protein, we notice that V1 is actually this cap that is covering the V2 and is actually masking it from the immune system. And so we hypothesize that the deletion of V1 could improve vaccine efficacy. We then established a collaboration with Professor Timothy Cardoso, which is a structural biologist, and we developed this new immunogen engineered, which is a GP120 deleting in V1, which has the V2 force in alpha helix conformation. And we applied this to our macaques as a DNA prime and ALVAC plus GP120 as a boost with delta V1 immunogens. And what we saw is that the V1 deletion in SIV envelope favoring this alpha helix conformation actually was better than our wild type. So what we did is then to use this vaccine and to understand what are the immune responses that are part of the mechanism of protection of this vaccine. And in 2023, I published this paper in Nature Communication in which I further analyzed the immune system and the innate system with myeloid cells. And what we identify is that the increase in CCL2 levels during the immunization and the engagement of classical monocytes is actually able to reprogram the monocytes from a pro-inflammatory M1 type to an anti-inflammatory M2 monocytes. And maintaining this balance is very important for HIV vaccines. So then we move forward and I further explore the possibility how these CD14 positive cells are important. And what I identify is that efferocytosis, which is a highly regulated process that is meant to eliminate apoptotic cells while maintaining anti-inflammatory environment, was not associated in the baseline CD14 collected from our macaques, but post-vaccination they were correlating the frequency of these efferocytes was correlating with the number of vaginal challenges required for the infection of the macaque, suggesting that the vaccination is inducing modification in these CD14 cells, and therefore they are more prone to conduct efferocytosis. We also identified other immunocorrelates of decreased and increased risk of HIV acquisition. In particular, analyzed the mucosal level of our macaques. HIV acquisition occurs mainly at the mucosal level. So we analyzed in our lab the innate lymphoid cells. And in particular, we identified two populations that were important. The NKp44 positive population were positively correlated with decreased risk of HIV acquisition, while pro-inflammatory, double negative, NKG2A negative, NKp44 negative ILCs that produce interferon gamma were negatively correlated with the decreased risk of HIV acquisition. So what we hypothesize through our all these analyses that I told you, but also other, is that following vaccination, we induce in our macaques anti-V2 antibody that mediate DCC, but also monocytes and CD14 positive cells that are anti-inflammatory. At the mucosal level, when we expose the animal vaccinated to the virus, some CD4 are of course getting infected, and what's happening is that the antibody against V2 together with NK cells eliminate these cells and they make them apoptotic. And the apoptotic cells are then cleared while maintaining an anti-inflammatory environment that doesn't recruit further target cells for the virus by the CD14 efferocytes. This was very important, but then we wanted to move forward, and I took on this project starting a couple of years ago at this point, in which we try to use a new adjuvant to further improve the vaccine efficacy. And we established a collaboration with Dr. Mangala Rao at the MHRP, the Walter Reed Hospital in Bethesda, because they have been developing since the 1980s different type of adjuvants that are based on these liposomes, the army liposome formulation called ALF. They developed a family of these adjuvants that include also the ALFQ, which includes on the surface of the liposome the QS21, which is the structure of several type of sugars, and this can be also used in association with the alum in already used in our protocols to absorb the antigen and merge them together to form the so-called ALFQA. So that's what we decide to use because of the importance of the alum in our vaccine. We use the ALFQA, and also because the ALFQ was a strongly potent liposomal vaccine adjuvant in rodents and non-human primates. ALFQA induced higher levels and more durable antibody levels. It's published by Mangala Rao in 2018. The ALFQ induced a balance Th1 and Th2 CD4 responses that are very important for HIV vaccination, and additionally these kind of adjuvants are already under testing in several, particularly 7 upcoming human safety and immunogenicity trials. So what I did in this project is to take some new naive animals and vaccinate them with our Delta V1 immunogens protocol, but with the boost of the protein GP120 Delta V1 in ALFQA rather than in alum. And I compared them to our previous vaccine with alum. When we calculated the vaccine efficacy, we saw that the alum has 60% vaccine efficacy, whereas the ALFQA had 79%, which seems to be higher. However, by comparing the acquisition curves of the two vaccinated groups, we don't see a really significant difference between the two groups. Also due to the fact that the study was not powered enough, we would need really a lot of animals to see a difference when we have such a similar vaccine efficacy, although the ALFQA may be more potent. I then explored different immunoresponses to understand how these were affecting the vaccine efficacy, and first of all I analyzed the antibody responses against V2. As expected, there was no antibody response against the V1 region, here in blue and in red also for both groups. But then when I wanted to analyze the V2 response, we saw that ALFQA group here, especially for peptide 26, had a higher antibody response against this region. And unfortunately, when we looked at this V2 specific ADCC, we didn't see a difference in the two groups, suggesting that maybe there are also other effector function mediated by the antibodies that may be involved, and that's what we are exploring right now, doing further analysis in cellular cytotoxicity. I move forward analyzing also the ferrocytic activity of the CD14 cells and how that was affected by the ALFQA. I performed again this efferocytosis assay in which we isolate CD14 positive cells from the vaccinated macaques, both a baseline and post-vaccination, and we color them with this cytotel blue, and then we co-culture them with colored and apoptotic neutrophils. And we look at the frequency of CD14 positive cells that are able to engulf these apoptotic neutrophils and at their engulfing capability. And what we saw is that the ALFQA actually increased the vaccine-induced frequency of these CD14 efferocytes, and not only, it increased also their ability to engulf apoptotic cells. So not only they are more, but they are also more prone to eat apoptotic cells. Finally, we wanted to understand if there was a difference in terms of the mucosal responses. And what we saw on the innate lymphoid cells in the mucosa is that the NKp44 positive ILC that I told you before, they are associated with protection in our previous study, and that they are important to maintain tissue homeostasis, were actually correlating here and then were higher in the ALFQA group. Whereas double negative and ILCs produces interferon gamma that are pro-inflammatory were less post-immunization and were less induced by the vaccination in using the ALFQA rather than the alum. Finally, we wanted to explore the proteome in the plasma of the two different groups, and what we saw at two different time points, 24 hours and 1 week following the last immunization, is that there was a clear separation between the two groups, immunized with alum and ALFQA, suggesting the importance of the role of the ALFQA. And one of the drivers of the difference between the two groups was the chemokine CCL2, which is actually important, as I told you before, to engage the CCR2 axis and to maintain a good balance between M1 and M2 monocytes. And as you can see here, there was a significant difference in the term of plasma CCL2 level at week 12 plus 24, which was higher in the ALFQA animals, suggesting that maybe it could induce a stronger activation of the CCR2 axis in monocytes that we are still exploring, of course, and unfortunately I don't have the data. So in conclusion, the formulation of the protein boosts in ALFQA resulting in a delayed SAV acquisition and high vaccine efficacy, and in increased antibody response targeting the V2, increased efferocytosis, increased NKp44 ILCs, and decreased negative interferon gamma producing double negative ILCs, and also increased CCL2 levels. So this is in conclusion what I did, but what is most important is also that now we are actually moving forward from the preclinical studies conducting non-human primates to human clinical trials, which will be testing in the clinical trial CLEAR, that is a collaborative study between the NCI and the NIH with Dr. Frank Maldarelli and Dr. Veffa Francini from the side of the NIH, but also with Dr. Mangala Rao, Colonel Julie Ake, and Merlin Robb from the MHRP. And this clinical trial will enroll roughly 90 volunteers that will be divided in 3 groups, in which one group in yellow will be the group with control, that is more RV144-like, and we want to prove that our vaccine strategy, both with ALFQA or hydrogel alum, are better than the RV144. And so we have also arm 1 and arm 2 with the DNA prime, followed by the use of Valvac expressing HIV genes, and the Delta V1 A244 GP120 formulated in alum or hydrogel. And then we will test different immunoresponses. And with that, I would like to thank everyone for their attention, and of course to thank all the people that took part to this very long and very important studies conducted at the NIH, and all the lab, of course, my mentor Dr. Francini, and all the other people that collaborated with us across the years. And with that, I would like also to thank you all and to take any question.